Okay, dear students, back to continuation part of recombinant DNA technology. Now move to continuation part of recombinant DNA technology. In the previous video, you have come across three techniques of recombinant DNA technology. Initially, I covered three steps. First is isolation of genetic material. First step is isolation of genetic material, that is DNA. Second, cutting DNA at specific locations. Third is amplification of gene of interest. Amplification of gene of interest using polymerase chain reaction. You know, it involves three steps: denaturation, annealing and primer extension. Denaturation, annealing and primer extension. In denaturation, target DNA is heated to about 94 degrees centigrade. Target DNA is heated to about 94 degrees centigrade. It results in the separation of the two strands of DNA due to breaking of hydrogen bonds. The two strands will behave like an template or master copy or blueprint. Second, oligonucleotide primers are used to hybridize. Oligonucleotide primers are used to hybridize the template strand from 3' ends. But this step requires low temperature, that is 40 to 60 degrees centigrade. Third, in primer extension, TAQ polymerase, Thermus aquaticus polymerase will synthesize the DNA. TAQ polymerase will synthesize the DNA complementary to that of template strand, but it requires deoxynucleoside triphosphate and Mg++. But the temperature which is required for the primer extension is 72 degrees centigrade. Here one interesting point: if the process of replication is repeated many times, if the process of replication of DNA is repeated many times, DNA can be amplified even approximately billion times, that is 1 billion are made. If the process of replication is repeated many times, okay, if the process of replication of the DNA is repeated many times, DNA can be amplified even approximately billion times, that is 1 billion. That amplified DNA is used in recombinant DNA technology. Followed by that, now dear students, cover three more steps related to recombinant DNA technology. Fourth is insertion of recombinant DNA into host cell. Insertion of recombinant DNA into host cell. As you know already, E. coli are frequently used as an host cell. E. coli are frequently used as an host cell. So there are several ways to introduce recombinant DNA into the host cell. So dear students, now look over the fourth point: insertion of recombinant DNA into the host cell. E. coli are frequently used as an host cell in biotechnology. E. coli are frequently used as a host cell in recombinant DNA technology or genetic engineering. Okay, I hope that my simple line is clear. Now move to, there are several ways there are several technologies are there to introduce this recombinant DNA into the host cell. There are several techniques are there to introduce this recombinant DNA into the host cell. Already you have come across that technologies. Number one, heat shock method. Second is micro injection, electroporation. gene gun. Even disarmed pathogen are also used as a vector to transfer the desired genes into plant and animal cell respectively. Okay? But now take care of one important point. When this recombinant DNA is having resistant gene, when the recombinant DNA is having resistant gene for antibiotic like ampicillin, okay, when the recombinant DNA is having resistant gene for antibiotic like ampicillin is introduced into the host cell, then host cell is transformed into ampicillin resistant cell. Look over the point carefully. There are several ways are there to introduce recombinant DNA into the host cell. For example, heat shock method, micro injection, electroporation, gene gun or biolistic, etc. In addition to that, now you have to take care of one more important matter. When the recombinant DNA is having resistant gene for antibiotics like ampicillin is transferred into the host cell, then host cell will become resistant to ampicillin, or host cell is converted into an ampicillin resistant cell. This transformed cell or recombinant cell, this recombinant cell can be easily isolated from the non-recombinant cells. It is easily isolated from the non-recombinant cell by spreading on a plate containing ampicillin. So look over my point. If the recombinant DNA is having resistant gene for antibiotics like ampicillin is transferred into the host cell like E. coli, host cell is converted into or it is transformed into ampicillin resistant cell. This ampicillin resistant cell or recombinant cell or transformed cell can be easily isolated from the non-recombinant cell or non-transformed cell by spreading on a plate containing ampicillin. Only recombinant cells will grow, but a non-recombinant cell will lose their life. I hope that my point is clear. When the recombinant and non-recombinant cells are spread on a plate containing ampicillin, only recombinant cells will grow, but whereas non-recombinant cell will lose their life or they will die. So it is another important point where you have to remember antibiotics like ampicillin, tetracycline, kanamycin will act as a selectable marker. This point already we have come across in PBR 322, plasmid of Bolivar and Rodriguez strain number 322, where I covered origin of replication, selectable marker and cloning site. So I hope that the fourth point is clear. Now move to next point, getting a foreign product, or we have to obtain the foreign product. Now look over next fifth point. So getting or obtaining a foreign gene product. Obtaining a foreign gene product. Okay. So here generally what happens, there are two simple technologies are there to get the foreign product. Before that you have to remember one important line. When the recombinant DNA is ligated to, plasmid or vector, then it is introduced into the cell. First it will establishes, then it will start synthesizing required protein. When the recombinant DNA is ligated to, vector, Then it is transferred to the cell. It will first establishes, then it will start synthesizing proteins. First it will get established, then it will start synthesizing protein. There are two simple methods they are following. Number one, when the biotechnologists want to produce the product in a small scale, when the biotechnologist want to produce the product in a small scale, then definitely they will follow laboratory culture. When the biotechnologist, or you can call it as a genetic engineers, has planned to produce the product in a small scale, they will 100% follow laboratory techniques or laboratory process. When they are planning to produce the product in a large scale, better go for continuous when you are planning to produce the product in a large scale, better go for continuous culture system. In continuous culture system, used material is drained out from one side and fresh material is introduced from another side. So look over dear students, if you are planning to produce the product in a large scale, better adopt continuous culture system. In continuous culture system, used material is drained out from one side. Used material is drained out from one side and fresh material is introduced from another side, so that you can keep the cell in the most physiological exponential phase. You can keep the cell in the most physiologically active phase, so that you can easily increase the products. If you are planning to produce the product in a large scale, better go for continuous culture system. In continuous culture system, used material is drained out from one side and fresh material is introduced from other side, so that you can keep the cell in the most physiologically active phase. Most physiologically active phase that will increase the production. Last point, downstream processing. This question has appeared many times for 1 mark. So very simple matter. So the product is separated. The product is separated and purified. The product is separated and purified. This process is called as downstream processing. The product is purified and separated. So even it is formulated with the preservatives. Before transported to market, it is formulated with preservative. When they are going to produce the drugs, now I am not talking with respect to narcotic drugs. You know already, the drugs which will affect the nervous system are called as narcotic drugs. The drugs which will affect the nervous system are called as narcotic drug. But here I am purely talking with respect to medicines. When they are going to produce the drugs based on the biotechnological idea, it is subjected to clinical trial, and even the quality is also checked by different methods. Quality is also checked by different methods. So that ends recombinant DNA technology. Total 6 points I discussed. Even we have to take care of diagrammatic representation. Students, look over this diagrammatic representation of recombinant DNA technology. This is diagrammatic representation of recombinant DNA technology. Once in the theory examination, they have arised this question for 3 marks. You have to draw a neat labeled diagram. At that time, explanation is not required. So many times I repeated, take care of the questions. You have to write what they are expecting. Diagrammatic representation means only figure you have to draw. Here look over the way of drawing the figure. Foreign DNA, okay, foreign DNA. Opposite side they have given vector. Under vector, many examples I quoted, but frequently plasmids are used. Isolation of pure DNA, followed by that it will come under the action of restriction endonucleases. Vectors are selected, then this vector is cut with the help of the same enzyme REN. Then this fragment of foreign DNA is ligated to plasmid with the help of an enzyme ligase. So look over here, first is isolation of pure DNA. Then it will come under the action of restriction endonucleases. You know already, REN will cut the DNA at the specific site having palindromic sequence. Vectors are selected, under that plasmids are frequently used. Then it is cut with the help of the same enzyme to produce sticky ends or single stranded ends or cohesive ends. Then the fragment of foreign DNA is connected to this vector with the help of an enzyme DNA ligase, and the DNA so produced is called as recombinant DNA, and this recombinant DNA plasmid is transferred to the cell where it will undergo replication and produces many copies. So you have to practice the figure, okay? So now look over, dear students, last matter related to first chapter of biotechnology, okay, principles and processes. Bioreactor is also called as fermenter. Bioreactor is also called as fermenter. Bioreactor is a container or a vessel. Bioreactor is a container or a vessel which are used to culture the cells at a controlled environmental condition for the production of some commercially valuable product. Look over the definition. Bioreactor is a container or a vessel which are used to culture the cells at a controlled environmental condition for the production of some commercially valuable product. Already I quoted a few examples for commercially valuable product. It includes antibiotics, DNA vaccines, enzymes, hormones, clotting factor for the hemophilic patients. Bioreactor is a container or a vessel which are used to culture the cells at a controlled environmental condition for the production of some commercially valuable products. Bioreactor are of various types. Here I listed 4 types: stirred tank bioreactor, fluidized bed bioreactor, packed bed bioreactor, airlift fermentation bioreactor. So bioreactor are of various types. Stirred tank bioreactor, fluidized bed bioreactor, packed bed bioreactor, airlift fermentation bioreactor. But for your syllabus, they have put forward stirred tank bioreactor. For your syllabus, they have put forward stirred tank bioreactor. Now look over first stirred tank bioreactor in detail. Only 4 to 5 points are there. Very simple matter. You have to take care of these matters because many times this question has appeared in the examination. Bioreactors of different capacity, have been designed depending upon their objective and requirements. Bioreactors of different capacity have been designed depending upon their objective and requirements. But bioreactor which are used in biotechnology, generally having the capacity of 100 to 1000 liters. It is an 100 to 1000 liter cylindrical metal container with curved base. So bioreactor which is used in biotechnology is having a capacity of 100 to 1000 liters. 100 to 1000 liter. No doubt it is made up of metal container and cylindrical in shape. Generally base is curved. Now question arises, what is the use of this curved base? It will facilitate the mixing of the content or it will facilitate mixing of materials. So it is made up of a very good quality metal with curved bases. That curved base will facilitate the mixing of material. Even it is provided with oxygen delivery system. It is provided with oxygen delivery system, pH control system, temperature control system and sampling port. Look over here. It is provided with oxygen delivery system, pH control system, temperature control system and a sampling port. Now look over first. It is having 6 flat blades. It is having 6 flat blades connected with motor. Agitation. It is having 6 flat blade connected to motor that is meant for agitation of material, mixing of the material to supply oxygen to all the cells. It is having stirrer shaft. First point, it is having a stirrer shaft with 6 flat blades. It is having stirrer shaft with 6 flat blades connected to motor and it is meant for agitation of material, mixing of material to supply oxygen to all the cells. Oxygen delivery system through which the air bubbles are supplied. pH control system or acid base for pH control. For example, when the pH of the bioreactor will decreases, alkalis are introduced through this port. When the pH of the hydrogen ion concentration of the bioreactor will decreases, alkalis are introduced through this acid base for pH control import or inlet. Port, in-port, or you can call it as a lid-like structure. It is also having temperature control system because enzymes will grow in a specific temperature, or the growth of the cell can be increased at a particular temperature, and it is having a sampling port. The sampling port helps to collect a small culture. It helps to collect the small culture periodically. It helps to collect the sample periodically. So for CET purpose, you have to remember 3 important points. For NEET examination or for CET purpose, you have to take care of these points. The term biotechnology was first coined by Carl Ehrlich, a Hungarian engineer in the year 1919. The term biotechnology was coined by Carl Ehrlich, a Hungarian engineer in the year 1990. Berg was often considered as a father of genetic engineer. Berg was often considered as an father of genetic engineering. Berg was often considered as a father of genetic engineer. Stanley Cohen and Herbert Boyer constructed first artificial recombinant DNA. Stanley Cohen and Herbert Boyer constructed first artificial recombinant DNA by using the plasmid of Salmonella Stanley Cohen and Herbert Boyer constructed first artificial recombinant DNA using the plasmid of Salmonella typhimurium. So like this, you have to prepare even the scientific words also.